Cells are the basic building blocks of all living things. A cell has a thin outer layer called the cell membrane. This membrane is like a gatekeeper that controls what goes in and out of the cell. It is made up of fats and special proteins called membrane proteins. These proteins are like tiny machines that do important jobs within the cell membrane. Membrane proteins are not evenly spread throughout the cell membrane. Some parts have more of a certain type of protein than others. This uneven distribution is called asymmetry, and it is important for the cell to function properly. Imagine a busy city with different gates for different purposes. Similarly, membrane proteins act as gatekeepers, transporters, and communicators for the cell. These proteins are essential for cells to interact with their surroundings and carry out vital functions. Membrane proteins are categorized into three main types: integral, peripheral, and lipid-anchored proteins. Each type interacts with the cell membrane differently. Integral proteins are permanently embedded within the cell membrane. Some integral proteins span the entire membrane, acting as channels for transporting molecules. Others are partially embedded, acting as receptors for signals from outside the cell. Peripheral proteins, on the other hand, are temporarily attached to the surface of the membrane. They can be easily separated from the membrane and are often involved in cell signaling. These proteins interact with either the integral proteins or the lipid molecules of the membrane. The third type, lipid-anchored proteins, are attached to the membrane through a lipid molecule. This lipid acts like an anchor, holding the protein close to the membrane. These proteins play crucial roles in signal transduction and cell adhesion. Integral membrane proteins are essential for many cellular processes. They serve as channels, allowing specific ions and molecules to pass through the otherwise impermeable cell membrane. Imagine them as bridges spanning a river, allowing only certain vehicles to cross. These proteins are also involved in cell signaling, relaying information from the outside environment to the inside of the cell. One of the key characteristics of integral proteins is their ability to interact with the hydrophobic core of the lipid bilayer. This interaction is facilitated by the presence of hydrophobic amino acids in the protein structure. These hydrophobic regions are attracted to the fatty acid tails of the lipid molecules, anchoring the protein within the membrane. Section 4. Studying membrane proteins, unveiling their secrets. Studying membrane proteins is crucial for understanding their functions and developing new drugs. However, these proteins are notoriously difficult to study due to their hydrophobic nature and complex interactions with the membrane. One technique used to study membrane proteins is freeze-fracture analysis. This technique involves freezing and fracturing the cell membrane, revealing the proteins embedded within. Another method is X-ray crystallography, which uses X-rays to determine the three-dimensional structure of proteins. Computational techniques, such as molecular dynamics simulations, are also increasingly used to study membrane proteins. These simulations use computer models to predict the movements and interactions of proteins within a simulated membrane environment. Section 5. Conclusion. The significance of membrane proteins. Membrane proteins are essential for life. They play vital roles in transporting molecules, cell signaling, and maintaining cell structure. Dysfunctional membrane proteins are implicated in many diseases, such as cystic fibrosis and Alzheimer's disease. Understanding how membrane proteins work is crucial for developing new treatments for these diseases. Researchers are continually developing new methods to study these complex proteins and unravel their secrets. The study of membrane proteins is a rapidly evolving field, and new discoveries are constantly being made. As we learn more about these tiny gatekeepers, we gain a deeper understanding of the fundamental processes that govern life itself.